people should hear this part of the conversation too. That I told you guys, try 2 is a doozy. There's a lot of information and a lot of lecture. So yeah. So we didn't make it to any of the questions and stuff that I had previously. What do you guys think is more beneficial for you guys? Is it more beneficial to kind of just go back through the material, or do you want me to dive into like question type practice? What do you think is more beneficial for you guys? A combination? Okay. Let me turn this notifications off.
I figure answering questions would probably lead to the going back through the material, probably.
Okay. Well, let's kind of plug through the material, and I'm going to kind of treat it similar to what we were doing before. I'm going to kind of come up with questions on the spot, ask you guys questions, kind of specify where, like, what kind of questions that you're most likely going to see. That way you get that that chunk of the material. Let's see really quick. I'm going to share my screen. Let me actually go to this one. There's one. Okay. So, actually, let me open this up as well. Okay, please. So last week we got through overall most of this. We got through the action potential stuff. We got through the excitation, the coupling, contraction, relaxation, muscle fatigue. We went over that. We read through the lab material stuff, and we covered enough of kind of the skeletal muscle fiber types, right? Is that all? Before we move on, any questions about last week's material? Anything that's still just like, this is not sitting right? Okay, then we'll keep going. Did you guys, because you guys had your lecture today, did you guys start with, what did you guys start with today? Did you go into smooth muscle? Did you jump into the heart stuff? Or was it the spinal cord reflex stuff?
Spinal cord reflex.
Okay.
Same for blended.
Okay. I love this stuff. This is the fun stuff.
Lily, come.
Okay. That all up. Okay, how comprehensive was this one? Decently comprehensive. I'm gonna pull up the other one as well, just so that we have it. There we go. These are probably my most favorite lessons of physiology, is the reflex stuff and the heart stuff. That was my favorite subject in gross anatomy as well, was all the heart stuff. Super cool. If you're in the live program, if you're in the on-campus program for gross, at some point you guys will do a total spinal cord laminectomy. So you remove all of the posterior portion of the vertebrae of the entire spinal cord, or the entire spine, and you get to see the entire spinal cord sitting in the inside of the spinal canal, which is pretty sick.
Yeah, I have lab tomorrow. Dr. G said that that's this week, so I'm kind of excited.
Nice. That was a really cool lab. After you do that, I think it's next chunk, you do a full craniotomy as well. So you remove the full cranium and you remove the brain as well. So really cool stuff. Okay. Starting out, let me look at our learning objectives, okay? Describe the anatomy of the spinal cord and its function in relation to the brain and spinal nerves. Describe the anatomy and physiology of the spinal reflex and reflex arc. Indicate the role of motor neurons and interneurons in the spinal reflex. And then give examples of spinal reflexes and their clinical significance. Okay, before going forward, we have two chunks of definitions. These are actually almost identical. I bet you he just copied Dr. Van. Yeah, it looks like he copied Dr. Van Winkle's slides almost perfectly on a lot of this. Yeah. Almost slide for slide. Okay. When we hear the word reflex, what does that mean to us? What comes to mind when we hear the word reflex?
Involuntary movement.
It's something that we do without thinking, right? Like, for example, I don't know how many of you have siblings, but if you have younger siblings, at some point, they annoyed the crap out of you. And they would poke you and poke you and poke you and poke you. And you get to the point where they go to poke you and you just automatically deflect them. That's kind of the reflex. We're going to talk about some specific reflexes in the body, but it's one where there's no need for cognitive processing. There's no need to think about it. Your body just does it. Okay? These specific, we're going to talk about very super specific, like spinal cord reflexes with this. But like you could extend this to as well, if anybody's ever heard the term muscle memory, where you do something so much, it just becomes muscle memory, becomes automatic. That's the reflexes that we're going to talk about, the spinal reflexes, are that muscle memory that's automatically built in to your body system, okay? The advantage to reflexes is that you don't have to think. Because even thinking, even if it's super, super quick, that those couple milliseconds or half a second, whatever it is, that it takes to think and to process and then to do something can be the difference between serious bodily injury and being perfectly fine. Kind of like, for example, if has anybody ever stepped on something really sharp? Has anybody stepped on a Lego before?
Yes.
Oh yeah.
Yeah. What do you do immediately when you step on something sharp or step on a Lego or a piece of glass or a nail?
Take your foot off of it as quickly as possible.
So yeah, so let's say your left foot, you step on that piece of Lego with full force, that foot comes up. What happens to the other leg?
Plants it.
It
Plants, right? It extends. So we have a flexor response on the side of the stimulus and an extensor response. So physiology, this is where we really get to start when we enter reflexes and this kind of stuff. This is where we want to start thinking real world experiences, because your legitimate experiences, if you stop and you think about these things that have happened to you, you know what's going to happen, and you can start, rather than memorizing all this stuff, you can work through it logically, as if you like whatever when these things were happening. Okay, so looking at learning objective number 1, describe the anatomy of the spinal cord. So big key points of the spinal cord. We have technically 3 horns, but we're only going to focus on 2 of them for this activity, or for the purpose of this lesson. So here in the very, very back, we have the dorsal horn, just like when we talk about the dorsum of anything, we talk about the back, dorsal fin, dorsum, it's the back of that spinal cord. Does anybody know what the dorsal horn is responsible for? We got any of that in?
Sensory.
Correct, yeah. So it's sensory. And when we talk about nerves, right, we have afferent versus efferent. Which of those two are our sensory nerves?
Afferent.
Good. Afferent equals sensory. They go to the brain. So they enter the dorsal horn of the spinal cord. Something along those lines is always a question. That the dorsal horn is the afferent or the sensory portion, okay? The anterior horn of the spinal cord, or the ventral horn, what's that responsible for?
Motor function.
Motor function, and it's efferent. Efferent is motor, and that's from the brain to the muscle. Okay? And the way that those travel and go through everything are through our nerve roots. Okay, we have a dorsal root and a ventral root, which come together to create our spinal nerve. And all of these nerves kind of lovingly travel together, and then they split off and go about and go to all the different places that they need to go. Okay? That's really about as far as we need to know a lot of the spinal cord anatomy. Okay? Any questions about the spinal cord anatomy? Sweet. So that's our whole first learning objective. The anatomy of the spinal cord, its function in relation to the brain, and the spinal nerve. So we see that the signal goes to the brain through the afferent, through the dorsal horn, and we get efferent from the brain to the muscle. Okay. Next part, describe the anatomy and physiology of the spinal reflex and the reflex arc. Before we go into that, we're going to jump kind of into just more general about the reflexes and follow all of this, okay? So there's different types of reflexes, and this is where there's a lot of anatomy. So we want, not of anatomy, a lot of vocabulary. So we want to make sure that we understand what these reflexes, like what all of these words mean, okay? Because the only ones that we're really going to talk about are these guys in this course. We need to know that there are other ones, but we're going to focus on our spinal reflexes, okay? These are the ones that are important for this specific class, okay? So there's a whole bunch of different groupings. This is important to understand. These are kind of together, so these can be combined together. For example, you can have a spinal reflex that is monosynaptic that is also somatic, and that it is innate. So like these, it's kind of like the subcategories. It can be innate or it can be acquired. From there, it can be two other things. Does that make sense? The little tree going down?
Yeah.
Okay. And really, try, guys, if you're at home or sitting around and you can participate, really, really participate. The more that you guys talk, confirm that you understand what's going on, the better off you're going to be. And if at any point you are like, what the hell is going on, please stop me, okay? Because everything that we're going to do, we're going to continue to build, okay? And if we don't understand it well enough to be able to explain it ourselves, you're not going to get the next chunk, and it's just going to waste your own time, okay? So if you have questions, please stop me. Even if you don't know what the question is, you just want to say, that makes no sense, I will figure out a different way to run through it to help it make sense, okay? So we look at this, break down some of the words. Innate reflexes. If anybody's ever heard of innate intelligence. You'll hear about that later at some point, either in FOC or in future, like more of the philosophy side of things. Innate means that it's something that we are born with. Okay, everybody has it. Or everybody's
Those
supposed
ones
to
like
have it.
Moro and like the other startle reflex.
Exactly. Yeah, those kind of reflex. Those are innate reflex. Those are genetic. Yeah. The blinking, the palmar grasp, the ones that we're born with, that we don't have to learn, we don't have to figure out, we don't have to do anything with. We have them. Okay. Acquired reflexes are a little bit different. They're conditioned reflex, vomiting, salivation. So it's kind of like everybody has their own favorite food, and if you smell a food that you really, really like, you start to salivate. Versus somebody else who maybe that same food is like disgusting to them, makes them want to vomit. So you see, those are different from person to person, whereas like the innate reflexes are the ones that everybody is going to have the same. We have somatic reflexes. These are going to control the skeletal muscles so that when you step on that nail and you jump back, you jump off that foot, that foot comes up and then the other foot straightens out. And even like, see, I even did it instinctively with my hand. Or like if you touch a hot stove, this hand comes back. And then if you actually watch people when they do that, kids when they do this, the first time that you touch a hot stove, you touch the hot stove, that hand curls up and the other hand shoots back. You get a flexion response and an opposite extension response. So that is called the withdrawal reflex, because you withdraw from the painful stimulus. That's one that we're going to talk about. The other one is the knee-jerk reflex. Knee-jerk reflex is the one where you go in and the doctor's checking your reflexes and he hits your knee with the hammer and the knee shoots forward. That's the knee-jerk reflex. Those are the main two that we talk about in this class to understand reflexes. Okay, and both of those are somatic, meaning skeletal muscle. Okay, somatic, that was one of our words that we needed to know previously. That is for our muscles. We also have visceral reflexes, which controls the actions of the smooth, cardiac muscles, the glands, all that kind of stuff. So for example, that learned reflex, that salivation is done by glands. So the acquired reflex, salivating, is an acquired reflex that is also visceral, or also known as autonomic. Same thing when you eat food. You ever, as soon as you eat food in the morning or drink that coffee or whatever, immediately you have to go to the bathroom? Or maybe not immediately, but pretty shortly after you feel like, your stomach starts to grumble, you have to go to the bathroom. That's the gastrocolic reflex. As soon as you have something enter your stomach, it sends out a signal to tell your body your stomach stretches, and it sends out signals to the brain saying like, hey, we need to make more room in the intestines, so then you have to go to the bathroom. These are all different reflexes. And we have a couple different types. We have what are called monosynaptic, and we have polysynaptic. Okay, mono meaning one. It means that there's only one synapse from the stimulus to the spinal cord, essentially. Okay, there might be extras other than that, but when we look at the specific reflex itself, we'll see when it's the monosynaptic versus polysynaptic reflexes, which might have a whole bunch of different places that it goes and it connects. Finally, we have cranial reflexes, like blinking, salivating, water the eyes, stuff like that. There's a reflex called the glabellar reflex, or if you tap right here on your glabella, it makes you blink. And if you keep doing it, then you stop blinking. Works better if somebody else does it to you. That's one of our cranial reflexes. And then we have the spinal reflexes, which we'll talk about those, withdrawal reflex and the knee jerk. Any question about the classification of reflexes? The most common question you'll get asked is about, is it mono or polysynaptic? I'm gonna double check on this, but I'm 99% certain knee jerk is monosynaptic, and I'm pretty sure withdrawal is also monosynaptic, but we're gonna double check that one because it's been a little while since I've looked at it and I want to be sure. Okay.
Knee jerk. He said knee jerk was monosynaptic. I remember him saying that for sure. And then withdrawal, it's in the slides. pretty sure it's poly, but I don't remember. He
The
did say
withdrawal
today, though.
reflex can occur in either the upper or lower limbs and is a polysynaptic reflex. There it is. It's polysynaptic. I love Google. Okay, so remember those. The one that's monosynaptic, knee jerk, polysynaptic, withdrawal. I promise that that's a question. It's always, because a lot of this other stuff is like super obvious. This is the one that's like testable material. Okay. Monosynaptic, we always use knee jerk. Polysynaptic is the withdrawal reflex. Always a test question. Do I want to talk about this yet? Yeah, we'll talk about this. So this is getting a little bit deeper into what is actually considered. I don't like the word classification. I think better here would be like reflex, or better kind of defining the reflex, kind of defining specifically what's involved within the reflex, okay? Because there's lots of different steps. We can look step by step what happens with a reflex. So we're going to start with the monosynaptic reflex. We're going to have a stimulus happen, and eventually with that stimulus, stepping on the Lego, touching the hot stove, getting hit with the hammer on the knee, all of those things are the stimulus, the thing that happens. All of the stimulus is going to elicit or create a response. When we talk about the reflex and the reflex arc, okay, the arc meaning the whole flow. See how this kind of looks like an arc or an arch? We are not going to include the stimulus or the response as part of the reflex arc. This is a test question. It's always a test question. The question is almost always, it's one of those put in order the reflex arc. Sometimes it's put in order the reflex arc of the knee jerk, or they'll put it with the withdrawal, like step by step, what happens, okay? And we need to make sure that we know and understand, because it's always going to be, there's going to be like 6 choices, and the very first choice is always going to have stimulus or response in it. We do not count the stimulus or the response as part of the reflex arc, okay? They're separate. They're what happens before and what happens after the reflex arc. Have I made that clear? Repeated it like 4 different ways, so hopefully that's clear.
Abundantly.
Perfect. So where do we start? We start at a receptor. Okay, what is a receptor's job? What does a receptor do?
Receives the signal.
Exactly. It's going to be the one that receives the signal, and it's its job to send that signal off towards the brain. Okay. And if we remember right, what do we call the fibers? What do we call the type of nerves that go to the brain? Afferent or efferent?
Afferent.
Afferent. I drill that into you guys because that's always included in questions, and one little letter difference completely changes the meaning of the question. Okay. So make sure you pay attention. Is it afferent or is it efferent? So the receptor. In this case, we're going to talk specifically about our spinal reflex. We'll talk about the, we'll talk about the muscle spindles. The muscle spindles focuses with the knee jerk reflex, mostly is what we're going to deal with. So our receptor is what's going to be checking and receiving the signal. For example, the muscle spindle. Specifically, we also call muscle spindles intrafusal fibers. Okay, we have, make it a little smaller. We have intrafusal and extrafusal fibers. Okay. Our extrafusal fibers are our normal muscle cells. these are those contractile cells. The intrafusal fibers, our receptor cells. Cool beans, cool beans. This is a lot of this stuff is just going to be straight definition, kind of memorization type stuff that you need to know kind of what correlates to what. These kind of processes, you really, really, really need to draw out multiple times and be able to draw them out very, very quickly. If you the quicker you can draw it and it be accurate, the better that you're going to be able to know it. Okay, so our receptor responds. We get muscle spindle. Specifically, we're going to have muscle stretch. Make sure. Sometimes I use a different word. I want to see if there's any. Yeah. It's muscle stretch. It can also be worded how much and how fast a muscle is lengthened or shortened. Okay. For the most of our reflexes, we're going to focus on how fast it's lengthened, because it's easier to hurt your muscle it getting too long too fast versus it getting too short or getting shorter. Okay. So the nerve, some sensory nerve that's attached to the muscle spindles that takes it all the way back to the brain are known as type 1A afferent nerve fibers. This is just going to be memorization type stuff. This is one of the examples. A lot of times he'll ask lots of questions about the different types. There's type 1A, type 1B. We'll get to those other ones in a little bit. So the basic gist is we have a receptor. It receives a signal. It sends the signal along a neuron to the spinal cord. Now this is a reflex, right? So we're skipping the brain. So instead of going up the spinal cord to the brain to process and then send out a signal, instead we immediately synapse with a somatic motor neuron, also known as an alpha motor neuron. The receptor sends a signal, it goes into the dorsal horn, connects in the anterior horn, and we get a signal that goes directly to the skeletal muscle to tell it what to do. We have our afferent and our efferent hits the target cell, and then we have that response, typically a contraction is what we're going to see typically. We're going to get into the actual specific details of like the actual knee-jerk reflex and stuff in a second. Any questions about this though? About kind of that step-by-step what happens? Okay. With polysynaptic reflexes, it's almost identical. The only difference is that there's an interneuron in between the sensory neuron and the alpha motor neuron within the spinal cord. Typically, an interneuron is responsible for it needs some kind of change of chemical mediator. It's because a lot of these sensory neurons, these type 1A, these afferent nerves, are going to release acetylcholine. And we know acetylcholine activates the other nerves, right? There are other chemicals that we're going to use, like glycine or GABA. Anybody ever heard of gabapentin?
He mentioned it in lab the other day.
Yeah.
Yeah, my dad actually takes it.
Currently on it.
Yep. Gabapentin, extremely common drug now. Used to treat like 90% of anxiety conditions, nerve pain conditions, depression, all sorts of stuff like that. And even like a whole bunch of other random stuff that's like shouldn't like be related to gabapentin, but it is for some reason. These are going to inactivate. Okay. I remember this, glycine, the I inactivates, and GABA. I just remember, you just need to remember that GABA. You can remember GABA has 2 A's, inactivate has 2 A's, but so does activate, so that might not work the best. But yeah, remember those two. You'll also want to remember glutamate. I remember that one because glutamate activates. I think it kind of sounds cool. Okay, those will come back up later. So like I was saying, the polysynaptic, very, very similar, almost identical, just it has interneurons in between. This can be one single interneuron, or you could literally have dozens of interneurons that mess with a whole bunch of random stuff to come out. We don't need to go in that far, though, because that's way above our pay grade. Here's that reflex arc from the receptor to the motor neuron to the effector, and then outside of here we have, the response. Outside of here we have the stimulus. Those are not part of the arc. This is the example of polysynaptic. If we remove the integration center, that interneuron, then we have a monosynaptic. So all of this with the integration center is a polysynaptic. Any questions up to this point? Sweet. Looks like this is where he adds all of the anatomy stuff. We talked about all of this. The gray matter in the center is where everything happens. The anterior versus, I guess he gets into a little other stuff. We'll keep talking here. Talk about this. Did he go into a whole thing about acetylcholine and norepinephrine, or did he not talk about that?
He did in lab, but he said you don't have to worry about it for lecture, but you do need to know it for lab.
Okay. There's a picture of this. It's not super complicated. There's just different acetylcholine and norepinephrine are big ones that we're going to use. So just kind of take some time to kind of look at this and how different things are divided and how it's used. Norepinephrine, epinephrine is also known as adrenaline. So the epinephrine or norepinephrine is adrenaline. So think about what kind of situations, what kind of things happen if we have a bunch of adrenaline in our system. Things go up, heart rate goes up, smooth muscle slows down, the stomach doesn't. The stomach, blood goes away from the stomach and the intestines. Heart rate increases, breathing increases, pupils dilate, all that kind of stuff. So when we talk about norepinephrine, epinephrine, that's where we're going to look at that. And that's sympathetic nervous system, our fight or flight, versus parasympathetic, just chilling with acetylcholine. All of this. Interneurons, not super important. I doubt he'd ask that question. We mentioned the extrafusal versus intrafusal. This is just reviewing on this. The extrafusal is the bulk of the muscle. It's all the stuff that's connected to the alpha motor neurons. The intrafusal fibers, those are the small ones. These are our muscle spindles. Okay. They're going to be two. Did he talk about Golgi tendon organs as well?
Yes.
Okay. I'm going to make sure that I follow all the same stuff that make sure I mention all the stuff that he's mentioning. Alpha motor neurons. We'll come back to gamma motor neurons as we explain the whole loop. We talked about alpha motor neurons. We've mentioned previously what a motor unit is. I'm going to highlight it again here. This is always a test question. What is the definition of a motor unit? A single alpha motor neuron and all of the muscle fibers it innervates. always a test question. Like I said, we'll come back to gamma motor neurons. That's not super important. I don't know why he would. Did he talk a lot about gamma motor neurons? Or did he kind of skip over this?
He talked a little bit about it in lecture.
Okay. We'll come back to kind of what they're. You really only need to know like what their purpose is. You don't need to know like nitty-gritty details and stuff. He does mention interneurons. This is always a test question. That interneurons are 30 times more numerous than anterior motor neurons and very highly excitable, and they are only located within the spinal cord. They're inside of the spinal cord. Those are going to be test questions, guaranteed. That's about it for interneurons. Renshaw cells, we'll come back to those. Man, his stuff, this is ordered really weird. Okay, I'm going to jump to big picture rather than doing all this nitty-gritty stuff. Let's jump to big picture and come back to all of this. Because we can, if we understand big picture, we can start to narrow down into the nitty-gritty. Okay, so we're going to start with our knee jerk reflex. We all know with the knee jerk reflex, doctor taps your knee with the little reflex hammer right on the tendon. And what happens? Our knee kicks out. So we're going to look at why this happens. When we hit the tendon with the hammer, essentially what we're doing is we're forcing that tendon to stretch and lengthen really, really quickly. Okay, so when we hit it, the tendon. Let me see. Actually, do this instead. Let me pull out. A blank PDF. Give me 1 sec. This will be easier. When I wish I was in person and I could just write on a whiteboard. Sweet. Okay. We have our knee jerk reflex. We're going to be jumping back and forth through things. We're going to talk our way through each of these components. So we have our stimulus, right? The tendon super quickly, the tendon, let's see, the tendon and the muscle super quickly lengthen. Okay? When they lengthen, with the change in length, we'll also say with stretch, the muscle spindle receptors activate. He might not have talked about this, but the way that muscle spindles and all these things work is that it's not that they're turned off or turned on. They're always on, but they change how much signal they fire. For example, if we're just sitting here, relaxed, my arm is flat or stretched out, relaxed, right? The muscle spindles in my bicep are going to be sending a constant signal. It's just bum, 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 slow and even paced signal. It's sending a slow signal saying, okay, just letting the brain know, hey, we're good, we're still good, we're still good, we're still in this position, nothing's changed, everything's fine. And then all of a sudden, let's say somebody grabs my wrist and yanks on it really, really fast, stretching my bicep tendon. When we get that super fast stretch, we get an increase in signal. So this is what it looks like when it's relaxed. We get signals versus as soon as it gets stretched, we get something like this, where it sends a bunch of signal really, really quickly in order to have everything happen that's supposed to happen. So this is nothing changing. Length is changing. Specifically, yeah, we say it like this, changing. Okay. That signal is going to travel on our type 1A afferent sensory nerve fibers. This is the main one that we focus on. I'm going to dive into a little bit more specifics again, because technically there are two different types within the muscle spindle, okay? Within the muscle spindle, and occasionally this comes up as a test question. Within the muscle spindle, there's kind of two different types of intrafusal fibers. There's a nuclear bag fiber and a nuclear chain fiber. Okay? This is kind of the subtext that is typically one test question, but it's not super important to the main idea, okay? So we have nuclear bag fibers. And we have nuclear chain fibers. Okay. The nuclear bag fibers have, we just focused, they have group 1A fiber innervation. Nuclear chain fibers have both group 1A and another called group 2 fiber innervation. It's going to be one test question. The test question most of the time looks something like, which of the following has only type 1A fiber innervation? Something like this, or which has both group 1A and group 2 fiber innervation. And then you're going to have, if it's just group 1A or type 1A, it's going to be the nuclear bag fibers. If it's group 1A and group 2, then it's the nuclear chain fibers. That's about as deep as really we want to be going. Okay? So it looks like he has a good chunk of information on here. If he adds, like, puts emphasis on anything else, write that down. But honestly, that's about what I wrote there. That's all you should really focus on with that. Okay? The main one that we're looking at is the type 1A. That's the main one that we focus on. Okay, back to our knee-jerk reflex. So those type 1A sensory nerve fibers, where do those go to? What's the next step? Where are those going to? Elizabeth, do you have an answer?
I thought it did, and then I looked at the picture and I wasn't sure. So.
Give us what you think. Give us what you think.
Well, because the type 1A is inside the intrafusal, like in the muscle spindle, and that's going to go to the afferent neuron,
Pause. Let's pause for back up real quick.
right?
The muscle spindle is the receptor,
Okay.
right?
Yeah.
It's made up of intrafusal fibers,
Yes.
which include the nuclear bag and the nuclear chain fibers. That's what makes up the muscle spindle.
So they're both in. Okay. So I guess that's where he confused me today, because I was like, wait, huh? So,
Yeah,
but
like,
so you have both, like, the nuclear bag and the other ones, the excuse me, nuclear chain inside the muscle spindle. So in the intrafusal, like it's
yeah, so like this right here, this guy, this is our muscle spindle right here. And the muscle spindle is kind of embedded within and connected to the actual extrafusal muscle fiber tissue like this. Does that make sense? How that
got
is?
it. Yes.
And inside of the muscle spindle, you have the nuclear bag fibers and the nuclear chain fibers that make up the muscle spindle.
Got it. Okay, that makes more sense now.
Yeah. And so when muscle stretches, when it changes, this is going to want to send a signal. It's going to activate it. And so coming down in here and wrapping around this, right? And then also wrapping around this is our, let me just type it because it's so much easier. Are our type 1A afferent nerve fibers. So the muscle spindle is going to get activated. And when it gets activated by the movement, that's then going to cause a signal to get sent along the type 1A afferent nerve fibers. So these fibers are just here kind of wrapped around, waiting for that muscle spindle for something to change, to pass on that message of change. Does that make sense? Does that make a little bit more, is that a little more clear?
Yes, thank you.
Sweet. And so it goes along that afferent nerve fiber. Let's continue this drawing. It goes and goes and goes and goes all the way up wherever it needs to go until it finally gets to the dorsal horn of the spinal cord. And it enters into the dorsal horn of the spinal cord, remembering the dorsal horn of the spinal cord, remembering that the dorsal horn and the anterior horn, this is our gray matter of the spinal cord, the little butterfly, it's our gray matter. Around it is our white matter. This inside is our gray matter. And it's immediately, so it's dorsal horn of the spinal cord, immediately connecting to an alpha motor neuron, also just known as a somatic neuron, located in the anterior horn of the spinal cord. Does all of that make sense? Yes, no, maybe.
Yeah, a little bit.
Okay.
Yeah, a little bit. Yeah.
So what it does, so that signal sends along. The whole purpose of those muscle spindle fibers is to, like I mentioned before, is to protect the muscle. Is if the muscle all of a sudden lengthens too quickly, we're in danger of tearing the muscle because the muscle's not prepared for that. So the body, what it does is it wants to shorten the muscle to protect it, to keep it from getting hurt, from getting overstretched. So that signal is sent along those type 1 afferent nerve fibers, immediately connects an alpha motor neuron, and it goes to specifically the quadriceps muscle. So that connects to it connects to the alpha motor neuron, and it takes it down. This is now our somatic motor neuron, also known as our alpha motor neuron. And it goes to the quads. And it literally tells the quads, hey, we need to tighten up to shorten the muscle so that to prevent it from getting injured. So quads are activated to prevent muscle injury. And actually, right here, right, this ends our reflex arc. Because remember, the response, the actual contraction, doesn't count as part of our reflex arc. But that is our full and total reflex of what happens. So when you get asked questions about the knee-jerk reflex, everything that I have written down here, this is what you need to know. Specifically when it talks about just like the reflex process, okay? We're going to add some extra things in here because a couple other things happen at the same time that are related to the reflex, but not actually part of the reflex. Does that make sense what I just said? That it's related to the reflex, but not part of the reflex.
Makes sense.
Yeah.
Okay. Well, it'll be a little clearer in a sec. Specifically, what we're talking about is this little piece right here. This second connection, right? So at the same time as our type 1A afferent sensory nerve fiber comes up to connect to the alpha motor neuron, it also connects to an interneuron, which is going to connect to the antagonist muscle group of the one that's going to be activated. What is the difference between an agonist and an antagonist muscle?
So your agonist is the one that's doing your concentric contraction, and your antagonist is your eccentric contraction, or your agonist is your prime mover and your antagonist is like your support.
Yeah, so the agonist can be that main muscle or one that's supporting and doing the same action. The antagonist muscle does the opposite of that muscle. So for example, the quadriceps extends the knee. So what the muscle that does the opposite is the hamstrings, which flexes the knee. So when we need, with this reflex, when we need the quadriceps to contract really quickly to prevent injury, the body also stops and thinks and says, oh, okay, if the quadriceps are going to contract, we also need the hamstrings to relax to allow the quadriceps to shorten. Because if we activate the quadriceps and the hamstrings both at the same time, what's going to happen to the knee?
It's going to
Is there going to be any movement at the knee?
No.
No, exactly. So if there's no movement at the knee, neither the muscles are shortening or lengthening, so we don't get actually any benefit from doing the reflex. So what happens is we have this inhibitory interneuron. Is going to act on the antagonist muscles alpha motor neuron. So it goes over and it literally tells the antagonist muscle, hey, you need to stay off, you need to relax to let the quadricep contract. You can do the same thing, this whole same process, thinking about it in the upper extremity, where if the bicep needs to contract, what needs to happen at the tricep?
Needs to relax.
Exactly, the antagonist muscle needs to relax. And that's the purpose of this secondary, this inhibitory interneuron little guy, okay? That whole process is most of what you need to know from this whole chunk of stuff. You'll get questions about, you know, like the muscle spindles specifically themselves, about the other like the specifics of the muscle spindle with the nuclear bag and the nuclear chain, or like the muscle spindles are made up of intrafusal fibers, that kind of stuff. But other than that, like, that's most of what we're going to talk about. Now, there's one other thing that we do need to talk about that I skipped over, and that is these little guys are gamma motor neurons. Okay? The gamma motor neurons, we're going to continue. This is, again, another kind of sidestep where it's not part of the reflex, but it's very, very important. Keep this back up. Keep this up again. After this whole thing has happened, and now our quadriceps have contracted, our muscles are in a different position, or a different position than they were originally started. There needs to be a way for the muscle spindles to change, to adapt to the current length and condition of the muscle. And that is through gamma motor neurons. Let me actually do this. I'm literally going to just reopen this. Let's see if it'll let me do that. Nice. I'm going to draw this whole thing out. So we have our muscle spindle. And when I say draw it out, this is what I want you guys to be able to do. We have our type 1A alpha motor neuron. Goes into the posterior portion of the dorsal horn. And it comes and it synapses onto our alpha motor neuron of right here of we have our lovely femur, beautiful. We have the quadriceps muscle right there, big Q, and then we're going to have our hamstring muscle right there. A little easier to draw just if you just do it in one color. And then we have an interneuron. This comes and binds to an interneuron, which binds to the alpha motor neuron of the hamstrings. And we know the quadriceps flexes, hamstrings relaxes. This is our type 1A sensory neuron. This is our muscle spindle. Bam, that's what we talked about at this point. Gamma motor neurons, we need the muscle spindle. Let's make this muscle spindle a little more accurate, where it's an intrafusal fiber amongst all of our extrafusal fibers. we need a way for this muscle spindle to stay taut at the same tension as the extrafusal fibers around it. So we have another connection with the gamma motor neuron. The gamma motor neuron comes back and innervates the muscle spindle fibers. It innervates specific fibers that are going to cause the muscle spindle to go to the exact same level of tension as the extrafusal fibers that are all around it. And that's all it does. Any questions about this whole picture, this whole process, everything that's going on here? Take that as a no. Couple quick questions. What is the purpose of the inhibitory interneuron connecting to the alpha motor neuron of the hamstrings?
To keep the hamstrings relaxed so that the quadriceps can contract.
What is the job of the gamma motor neuron?
Maintain muscle spindle tension.
Yeah, to match the extrafusal fibers as well, so it continues to give the correct signal. Type 1A sensory neurons come from what two fibers located within the muscle spindle?
Say that again.
Type 1A afferent sensory neurons come from, or I should say, innervate what two types of fibers in the muscle spindle?
The nuclear bag fibers and the nuclear chain fibers.
Are the nuclear bags or the nuclear chains duly innervated?
Chain.
With what are the two types of neurons that innervate the nuclear chain?
So it's the
Type 1A and group 2.
yep.
Nice. Type 1 and group 2. Perfect. What are the two neurotransmitters that are released within the spinal cord that inactivate alpha motor neurons?
GABA and glycine.
Good. What are some of the ones that activate? What are some of the neurotransmitters that activate within the spinal cord?
Acetylcholine.
What's the other one?
Acetylcholine and glutamate.
Yeah.
Nice. How do we feel about the knee-jerk reflex?
Better.
Is the knee-jerk reflex mono or polysynaptic?
Mono.
Mono.
Good. What are the what is it? I think what are the steps of the reflex arc of the knee-jerk reflex?
Tendon stretch, tendon and muscle stretch. Then action potential to the spinal cord. Synapse with alpha motor neuron. Efferent neuron sends your signal to muscle, and then you get the contraction.
Very, very, very close. Everything that you said is technically correct. The question that I asked was the steps of the reflex arc.
Oh,
So
so
what
it
is
doesn't
not
include the
included?
stimulus or the reaction or the contraction.
Perfect. Does not include the stimulus or the response. Sick. Good job. What other nonsense do we got in here? I think it says it also has the stretch reflex. Not a lot to the stretch reflex other than dynamic and static. So dynamic is moving. That's where, you have a load and you feel how much the load is, and whether you want to move it or maintain it, your body adjusts specifically to what you're supposed to have. And if you add load, like if I'm holding my phone and then all of a sudden add load, there's gonna be a little initial drop, and then my body's gonna respond, have a reflex to automatically increase the number of muscles contracting, the number of muscle fibers contracting in order to hold up the new load. There's not like a ton to that. I don't know why he has he's got quite a few slides on it, which I guess if he spent more time on this, know it. Looks like it's another monosynaptic reflex. So just increase afferent signals. So there's more stress going on, more muscle spindle activation. So you get a stronger muscle contraction. That's a lot of slides just to say that. This is going to come up a couple of times, the muscle spindle. So one of the jobs of more the concept, just that one of the jobs of the muscle spindles is to smooth out the muscle contractions. So it changes and adapts that proper signal to allow us to perform smooth movements rather than doing a movement, having to reset, move, move like that. The muscle spindles, because they're constantly sending Oh, I get what he means here. He says signals are not constant, being that, where's the other picture like I talked about before? You get the action potentials sending at a rate. They're not constant because it's not one continuous signal. There's signal, then signal, then signal, then signal, then signal. But it is it is continuously signaling, but not constantly signaling.
Signaling at a constant rate.
Yeah. Words like word specifics get confusing with that one. But just understand the level of question that you're more than likely to get from this is he's either going to ask you word for word this right here: signals are dynamic due to sensations at muscle spindles, or it's going to literally be which of the following is responsible for smoothing out the muscle contractions or for preventing jerkiness, anything like that. And the answer is going to be the muscle spindles smooth out the muscle contractions. That can be a question that he can ask as well. Alpha gamma coactivation. This is, I already explained this going on with with the knee-jerk reflex, how when the alpha motor neurons activate and the muscle contracts, we have the gamma motor neuron that comes and innervates the intrafusal fibers to activate those as well. That's all that's referring to. It's just the specific name for it. Alpha gamma coactivation. They activate together. If the alpha motor neuron is activating, the gamma motor neuron also activates in order to maintain the signal. Just giving a specific name to what we described earlier. He might ask a question on this stuff right here. This is just more little detail stuff of the muscle spindles, where if you have a relaxed muscle, the spindles are really sensitive to stretch. If the muscle is fully contracted, this is just an example. If there's no spindle coactivation, this is just going to go back over. If there's not that gamma activation along with the alpha motor neuron activation, then your muscles aren't sensitive to stretch, because this the muscle spindle isn't at the same tension as the rest of the extrafusal fibers. And then the normal contracted muscle in a normal situation, you have the spindle coactivation, the alpha gamma coactivation, so that it remains sensitive to stretch of the muscle in order to, continue to prevent injury. There's all the knee jerk stuff. He gets in a little bit more detail. What we talked about with the inhibiting the antagonist muscle, that's known as reciprocal inhibition. I don't love to use all the big fancy words, but you might see them on the test. So, okay. Anything up to this point? Confusing questions, concerns?
My brain definitely hurts after it, but it's okay.
Yeah. My biggest piece of advice is take the time to draw it. Draw it out. Draw it a couple of times. It does not have to look pretty, and don't save it. I would have a notebook on my desk where I would take this and I would draw it, and I would say it out loud. Okay, there's a muscle spindle which has this, this, and this. A type 1A sensory neuron goes into this part of the spinal cord. It connects to the alpha motor neuron, which goes to this, which completes the reflex arc. And then you have, what's it called? The reciprocal inhibition. You have reciprocal inhibition of the hamstrings doing this. And then you have the gamma motor neuron, which goes and connects to this part of the intrafusal fibers in order to maintain whatever. And like going through it super, super quick like that, drawing it a handful of times, helps it really to stick. And then I would draw it once, really neat and nice, to have with like my notes and with all of my stuff. Couple of extra details. Clonus. Clonus is pathology. It's basically that it's an uncontrolled oscillation of muscle movements. Let me show an example of it. I'm going to get a little bit ahead into some of your pathologies. So see how the ankle, the foot is just thumping along. This is really rare that you will see something like this. What this is referring to, you have a couple different things that you can have happen with nerve and spinal cord type injuries. So this is above your pay grade, but I'm going to introduce it to you guys just that way you've heard it, so that way in the future you know what's going on. You'll have what are called upper motor neuron lesions and lower motor neuron lesions. They write them upper motor neuron lesions, and lower motor neuron lesions. Lower motor neuron lesions is anything that's spinal nerves out distally. Upper motor neuron lesions is CNS, so brain and spinal cord. And you have specific things and specific characteristics of both of these. Clonus is an example of something that occurs with an upper motor neuron lesion. So think of like a spinal cord injury. Think of if there's like a posterior central herniated disc that pushes on the spinal cord, not on the nerve roots, but on the spinal cord, you could have clonus. If you have like a spondy, you guys talked about spondies before? Thank you. It's been mentioned.
She mentioned it in imaging some, and then I know a little more about it, but
So spondy, you also call it anterolisthesis, is when you get kind of this slippage forward of a vertebrae on top of another vertebrae, like this. Way more common than you think. I've seen almost 24 already at this point of these guys. So because this can pull the spinal cord forward, and you can have the spinal cord hit on one of these. So you have spinal cord injury, stuff with that. But yeah, you'll get clonus. So clonus is just the muscle spindle. Essentially, it isn't working because it can't go into the spinal cord. The area where it's supposed to go into the spinal cord and get sensed is damaged, so it can't do its job. So you get clonus. Golgi tendon organs. So big key thing to remember. Muscle spindles deal with the stretch or the length of muscle. Okay. Golgi tendon organs deal with the tension on muscle. Okay. That's always a test question. Though one of those two ways, comparing Golgi tendon organ to the muscle spindles. They'll say which of the following is responsible for measuring tension on the muscle. Which of this following is responsible for measuring stretch or changes in length of the muscle? Okay, so Golgi tendon organ, the tendon measures tension. Okay, the spindle is length. Location of the Golgi tendon organ, they're located where it's changing from the muscle belly more into the tendon itself. That's why it's the tendon organ. And it causes relaxation of muscles, stimulating the nerve endings. So have you ever had like a really bad Charlie horse or something, and somebody helps like push and helps stretch out your leg or stretch something out? Has anybody ever had that experience?
Yeah.
yes.
Yeah. So what that's actually doing is taking advantage of the Golgi tendon organ. So what you do is when something's cramping or if you need to stretch something, you put it into its full stretched phase and you stretch it as far as you go. And if you hold it there, after a couple of seconds, it feels like the muscle relaxes and you can go a little further. And then the muscle relaxes again after a little bit and you can go further. Right? Have you ever felt that?
Yep.
That's that's the Golgi tendon organs working their magic. That's what they do.
So
Golgi.
Go ahead.
What was that?
No, I was just gonna say you kind of answered this, and this is maybe a little bit deeper than this class, but Golgi tendon organ. So basically, static stretching. That's what you're working on, right?
You can do that one way. There is another treatment, and I highly recommend. I'm gonna try and give you guys these clinical nuggets. There's something called PNF stretching.
That's what I was about to ask about too. Was like, is it PNF? Is PNF more Golgi tendon, or is static more Golgi tendon?
It's both. It's
Okay.
just PNF is more aggressive activation of it, because at the same time as you are, because you get like the static stretching increases tension on the muscle, but then if you add into that muscle activation, that takes the tension on the muscle to an even higher level. So it's because you can do static stretching, and then PNF stretching is like the step above.
Okay. Yeah, because like clinically, I've always found PNF to be more successful.
Yeah, it is more successful, and there's a lot of other reasons behind that has to do more with like combining like nerve activation and firing the nerves at the same time in order for like the brain to kind of assimilate and like also push, and you also get a lot of like fear avoidance type stuff, and there's a lot more like psychology and also like neurology type stuff involved with like PNF stretching and why it works better. So, but yeah.
Okay, thank you. Yeah, that was just a note I wrote on like during lecture today, but I wanted to ask you because he's not that kind of a doc.
Yeah.
So.
If anybody doesn't know, PNF stretching is where you bring the muscle to tension, kind of where it starts to get uncomfortable, and then you maximally contract the muscles, trying to activate them, and then holding it for a couple seconds, essentially fatiguing the muscle, and then bringing it further into the stretch and doing that 3 to 4 to 5 times. So it works really, really well for hamstring issues, for piriformis type stuff, sciatic type pain. Really, really, really good for those kind of things. File that away in your bank. We've already kind of mentioned a handful of these things, where we have, you know, the withdrawal reflex. It's that stimulated mostly by pain. It's polysynaptic, which means it has lots of interneurons. The flexor muscles are stimulated and extensor muscles inhibited on the same side. Let me add that in there. On the same side as the pain. So you touch the hot stove with your hand, flexors activate, extensors relax, and then you have what's called the crossed extensor reflex, where the opposite side, the opposite happens. So my biceps contract, triceps relax, biceps relax, triceps contract, to get away from the hot thing or from the Lego or anything like that. And again, this is really quick. You know, as you step on the Lego, you jump off of it, and then quickly after that, you're able to put your foot back down, figure it out. That's really all. And like the steps for this whole reflex works very, very similarly. Only difference is that instead of a muscle spindle, you have a nociceptor. I don't think he goes into this at all, but you're going to have C fibers. C fibers are your pain fibers or your nociceptive fibers. So you have your nociceptor, which is your pain receptor, which goes up the C fibers into the dorsal horn, which connects to a whole bunch of interneurons, which then, you know, you have the activation of one of the alpha motor neurons, inactivation or inhibition of the other alpha motor neuron, and then you have it go across, where you get the crossed extensor reflex as well. So like if you understand the knee jerk reflex really, really well, the rest of this is pretty simple, because as well, on all of this at the same time, you're still getting as well like alpha gamma coactivation type stuff, where because the alpha motor neuron of the biceps is being activated, you're also getting coactivation with gamma neurons to go and make sure that the muscle spindles are the right size. So any questions about the withdrawal reflex? I know we super skipped over it, but after going deeper into the kind of the knee-jerk reflex, I feel like this is a little more comprehensible.
It's understandable for sure.
Okay. Any other quick? Did you guys just do this PowerPoint today? Or
We
did you-
got into the excitation to smooth muscle.
Yeah. I think we got about halfway through it.
halfway
Looks
through
like-
smooth muscle stuff?
Yeah.
Like 8 slides in is where I stopped.
Same.
Well, let's do. I think I saved it here. Just a heads up, y'all's first exam is gonna be coming up pretty soon.
Tuesday.
Yep, Tuesday.
Yeah. If you check out the YouTube channel, so we're not gonna get to smooth muscle this week. I already have all videos up. I have videos up for smooth muscle and for the heart stuff. So I don't have everything perfect for all of this stuff that I have right now, but if you want to come back and rewatch this, you can. But yeah, so all of it is there. It's not as nicely broken up as this, but it is there. So if you want to go, if you need help in watching, like figuring that stuff out, that material is there for you. Who's ready for practice questions?
Always.
Woohoo!
Always.
These are pretty straightforward. Which of the following muscles is under voluntary control?
Skeletal.
Skeletal muscle, good job. Which are under involuntary control?
Cardiac and smooth.
Good. Which type of muscle is striated and multinucleated?
Skeletal.
Skeletal.
Skeletal muscle again. How many nuclei does cardiac muscle have?
1.
One, occasionally 2. What is the shape that we typically associate with cardiac muscle?
Striated, but it's like kind of back and forth. Yeah.
We call it striated. We also call it Y-shaped. Sometimes you
Yeah.
see it as described as Y-shaped. You guys haven't gotten to the heart stuff yet, so you'll talk about this more, but you'll talk about like intercalated discs and that kind of stuff, and gap junctions and all sorts of stuff later this week. Make sure you watch the videos on that kind of stuff. And if y'all want to, we can maybe do a session this weekend for test prep.
That would be wonderful.
Okay. Which type of muscle is capable of autorhythmicity or self-excitation?
Cardiac and smooth.
It's just technically cardiac. Smooth muscle it's not self-excitation, but it does have like continuous excitation due to stretch. If you stretch smooth muscle, it excites, but self-excitation, autorhythmicity is just cardiac muscle.
So is that
Smooth
because
muscle. Go ahead.
sorry, JC. Is that so like digestion, for example? Is it because of the stretch and that's in the smooth muscle in your digestive tract? Like is that kind of
Kind of as they call it the
Kind of like a
bolus.
stimulus. Like it's not
Mhm.
really self-excitatory, but it needs a stimulus to be excited.
Yeah, it has a stretch response.
Yeah.
So
Makes
smooth
sense.
muscle, like in the intestines, if it gets stretched, that stretching, I don't remember the exact process by which it occurs, but it's like the stretch, it causes mechanical release of calcium which then causes, the whole process to occur. So, which type of muscle is found in the walls of hollow organs, such as the stomach and the intestines?
Smooth.
Our smooth muscle. Which type of muscle has the slowest contraction and relaxation times?
Skeletal. Technically smooth. It's smooth. My
Yeah,
bad.
it's going to be
Yeah.
smooth.
I forgot that there's a chart in here
Remember,
that I was looking at.
smooth muscle are long contraction / relaxation times and very fatigue resistant. So the smooth muscle, if you think about it, the smooth muscle is going to be a really slow contraction because we don't need to rocket food through our intestines, right? We want it to take its time just so we can absorb all the nutrients and break it down and do the stuff that we need to do. But smooth muscle has very long, sustained, that's a word that you'll see, sustained contractions.
Yeah, he had
That's
a
the
good
word you'll
chart in
see.
here, but he kind of just talked about it for like just a minute and then he went on.
We'll look at it as well. Yeah, I'm sure. I trying to remember if I look at his notes for smooth muscle stuff or if I use more Van Winkle stuff, but I have the video on smooth muscle, so if you want to watch it, I recommend doing so.
Will do.
What is the basic functional unit of a muscle called? Sarcomere, myofibril, myosin, or actin?
Sarcomere.
Yeah.
Yeah. What's a myofibril?
An individual muscle fiber itself. Individual tensile unit.
Isn't it myosin and actin together?
Myosin and actin, they're both myofibrils.
Yeah.
Yeah. So remember, make sure you remember this next question. The orders of tissue layers. Which of the following connective tissue layers surrounds the entire muscle?
Endomyosin or epimyosin.
So what does endo mean? Or what does epi mean?
Inside. Epi's the outside.
Yeah, endo is inside. So deep. Epimysium is outside. Good. So it's epimysium.
Harry, it's around the fascicle, right?
What did you say?
Perimysium's around the fascicle, is that right?
Yeah.
Yeah.
That is correct. Endomysium, perimysium. So endomysium is the whole muscle. Perimysium is around the fascicle. And then endomysium is around the
Individual
muscle fibers.
Yeah.
A bundle of muscle fibers within a muscle is also called a sarcomere, myofibril, fascicle, or tendon.
Fascicle.
Yes.
Yeah. Teachers like to ask about fascicle. That's for some reason, that's one that students always get confused on, and a lot of teachers love to throw questions about fascicles in there. Also because it's the middle part. Most people know the beginning and the end, like the outside of the muscle, the inside of the muscle. But for some reason, fascicle is one that's hard to remember. Which of the following proteins is primarily responsible for muscle contraction? I don't love this question, but
Myosin?
yeah, myosin is correct because myosin is responsible for the power stroke. Myosin also is one that has the intrinsic ATPase, right? Actin, what is actin made up of? What are the smaller components of actin? How do we label those? I'll give you a hint. One is
Drake.
G-actin. Remember, G-actin is the individual little balls, and then put a small string of them together, and we get F-actin. And then all the F-actins combined together gives us the full thing of actin. Troponin. Troponin has 3 subunits. What are the 3 subunits? I believe in you guys. You can do this. What's troponin doing? What's its job? What's it holding on to?
I know it like receives the calcium so that tropomyosin moves off of the myosin binding sites.
Yeah, so the troponin has 3 jobs, right? It accepts calcium into the troponin C, and it holds on to the tropomyosin. Its troponin T holds on to the tropomyosin, and then it holds on to one other thing. What else does it hold on to? What's the tropomyosin connected to? What is the tropomyosin wrapped around?
The actin.
The actin. Or what's the letter that we use to represent actin?
Is that like the I band and the sarcomere?
I band.
We use I, yeah.
Yeah.
Good, because the I band of the sarcomere is the area that only has actin in it. And when we have a concentric muscle contraction, what happens to the I band?
Shortens.
It decreases. Good. Pulling some deep thoughts from the brain. Tropomyosin. What's tropomyosin do?
It covers the myosin binding sites on the actin until the calcium binds to the troponin, so then it moves and the myosin can attach.
Boom, perfect. Which structure stores calcium ions within a muscle cell?
Sarcoplasmic reticulum.
Yeah, specifically, what is the name of the protein within the sarcoplasmic reticulum that holds on to the calcium?
Calcitonin. Ooh.
That's the question. Good.
Like,
Great
that's
job.
completely Greek.
I spelt it wrong, but how do you spell it? Guys, I hate spelling calsequestrin. It's IN, that's what it is. Calsequestrin. Bam. The T-tubule. What's the T-tubule's job, guys? Oh, hey, look.
T-tubule is what brings it back, or it like sends it through the membrane, right?
Yeah, it conducts action potentials deep into the muscle fibers. Sick. Yeah. What are the 2 things on the side of each of the T-tubules called?
It's 2 different receptors.
More broad.
More broad than that? Oh, I just lost the word. I had it.
2 words. First word, terminal. Second word, cisternae. Terminal cisternae, which is just a fancy way of saying the end part of the sarcoplasmic reticulum. So he can say that. So, and it's, remember, those 3 things combined. So it's part of the triad, which is the T-tubule and 2 terminal cisternae, or you can also call it terminal sarcoplasmic reticulum. There's another question I was going to ask within that. Oh, thinking deeper, within the sarcoplasmic reticulum, especially like within the terminal cisternae, what are the name of the mechanoreceptors responsible for the release of calcium?
Ryanodine one?
Yep. Perfect, the ryanodine. So we have the ryanodine, which are our mechanoreceptors. That's typically a test question. If it's not that one, what are the voltage-gated receptors?
It's like DHP, but I don't remember what that stands for.
It is DHP, dihydro...
Pyridine.
Yeah, pyridine, something like that. Those are voltage receptors. How does calcium get back into the sarcoplasmic reticulum?
ATP.
Through ATP, what's the name of the protein that uses the ATP?
The calcium ATPase?
Yeah, calcium ATPase pump. Sometimes it'll just be calcium pump, but it uses ATP. If we use ATP, what kind of transport is that?
Active transport.
Active. And is it diffuse or is it facilitated?
Well, it's I feel like I always screw these up. I want to say facilitated.
Facilitated.
If
What
you
does
need
facilitated
ATP,
mean?
then it's facilitated. Like it needs energy to move.
So if it needs energy, it's active.
Okay.
If it's facilitated, what does facilitated mean? Really making you guys think back.
This is where I get these mixed up. Because facilitated, I always want to think active because it's like helped along. Because like facilitate, like if you facilitate something, then you like move it along.
So this is kind of one of the all squares are rectangles, but not all rectangles are squares. All active transport is going to be facilitated, but not all facilitated transport is going to be active, because facilitated just means that it uses a protein, an intermembranous protein. So that could be like the calcium ATPase pump, the sodium potassium pump, or it could just be one of the ion channels. It could be one of the voltage-gated ion channels. It could be one of the leaky channels. It could just be one of the what's it called? The aquaporin? I'm saying that wrong, but one of like the water channels, that just lets stuff pass through. Facilitated means that it uses an intermembranous protein or a channel of some kind.
And diffusion's like it's not going against the concentration gradient, right?
Yeah, exactly.
Okay.
What's the difference between diffusion and osmosis?
Osmosis is water.
Just water, yeah.
Yeah. Osmosis, water is moving. Diffusion, stuff is moving. Which of the following describes the sliding filament theory of muscle contraction?
A. So actin filaments slide over myosin filaments to cause contraction.
Good. Does that make sense to everybody? Why that's the answer? Once the myosin the action of the myosin head binding and pulling, what is that called in the sliding filament theory?
Like the. So that's like the power stroke phase. Is that what you mean?
Exactly. Yeah, that's what I meant. The power stroke. Dope. The region of a sarcomere where actin and myosin filaments overlap is called the blank.
Z-band?
Z-band is not an answer.
Z line's on the outside.
Right, Z line is the
Is it the
outside, right? Because
Yeah, it's the
that's
outside
the
one.
end line of the sarcomere. Remember, because a sarcomere measures Z line to Z line.
Oh, that's right.
And it's approximately 2.2
Micro. Yeah.
Micro something. I don't remember if it's micrometers or I think it's micrometers. Think.
Is it the A band though? Like, is that the answer?
Yes, it is. A band is the correct answer.
A is for dark. That's how I get that, because it's dark whenever you look at it through like a.
Yeah.
I apparently need to go through the sarcomere again because.
Oh yeah, this is another one. It helps to draw it. It's super simple like this, right? We have the I band, which is all actin. We have the A band, which is myosin and actin. We have the H zone, which is just the myosin. And we have the Z lines. And what's that fancy little protein that connects to the myosin that helps with elasticity?
Titin.
Yeah,
What's Titan? It's called Titin, whatever. Also known as connectin.
Connectin.
Yeah, so if we look at this, when this contracts, of all of these, which of the following is going to remain the exact same size no matter what's happening?
The I band, Z line, right?
The Z line will remain the same. Yeah,
Yeah.
technically. Which
the
of the
A
bands?
band, right?
Yeah, the A band is going to remain the same because it's the myosin and the actin. If we have concentric contraction, which means the muscle is shortening, the H zone and the I band are both going to get smaller and potentially disappear. If we have eccentric muscle contraction, which kind of sounds like an oxymoron, eccentric muscle contraction means that the muscle is active, it's contracting, but it's lengthening. If it's lengthening, we get an increase in the H zone and the I bands. Which of the following is the correct order of structures from largest to smallest?
C.
Good job. What's another name for a muscle fiber? Myocyte or muscle cell. What about myofibril? What if we split?
Myosin.
Yeah, what if we split myofibril into two? What would we put from largest to smallest?
Largest to smallest would be myosin and actin.
Good. Which of the following statements about muscle contraction is true? The length of the sarcomere remains constant during contraction. Muscle contraction is initiated by the release of calcium ions from the sarcoplasmic reticulum. Actin filaments shorten during muscle contraction. Myosin heads bind to tropomyosin during muscle relaxation.
Initiated by the release of calcium ions. B.
Good. So we know that the myosin heads do not bind to tropomyosin, but how do the myosin heads release from the myosin binding sites?
Your ATP binding to it.
if somebody dies and no longer is producing ATP, what is it called when you get locking up or sustained contraction of the muscles without any ATP?
Rigor mortis.
Yeah. Which structure connects muscle to bone?
Tendon.
Tendon. What does ligament connect to?
Bone to bone.
Yeah. Please remember that. Please remember that. Little clinical nugget. What is it called when you injure a muscle? Let's see. Pull a muscle.
Strain.
A strain. What about if you hurt a ligament?
Sprain.
Sprain. Good. Which type of muscle fiber is primarily used for endurance activities?
Type 1.
Yeah. Which of the muscle fiber types is going to be used for sprinting?
2B.
Yeah, or type 2B. A big beefy bodybuilder. What are they gonna most have what muscle fiber type is going to be the most, let's say, not technically true, but we'll say the most prevalent?
2A.
Either 2A or 2B, one of the type 2 muscle fibers, yeah. Which of the following is not a function of skeletal muscle? Produce heat, maintain posture, produce movement, or store calcium?
I was gonna say store calcium, but I feel like that's
right. No, that's right. That is not a function of skeletal muscle. It is not responsible for storing calcium. That is what our bones are for. This is actually why start as soon as you can. They recommend weightlifting at least 2 times per week or doing some kind of heavy impact movement or exercise, be that sprinting, jumping, something along those lines, to promote strong bones and to not lose bone density, because once you lose bone density after you've gone through puberty, you do not get it back. The neuromuscular junction is the connection between a motor neuron and a blank. Sarcomere, myofibril, muscle fiber, or tendon?
Sarcomere?
Motor neuron. We have motor unit. What's a motor unit?
The motor neuron and all of the muscle fibers it innervates.
Muscle fibers, right? Sarcomeres are smaller than muscle fibers. We don't innervate individual sarcomeres. We innervate the muscle cells, which are AKA the muscle fibers. I was gonna ask another question about this. What voltage gated ion channels are responsible for the release of acetylcholine into the neuromuscular junction? Want me to repeat the question?
Yes, please.
what kind of voltage-gated ion channels are responsible for the release of acetylcholine into the neuromuscular junction?
Is that the cation one? That's not like the whole name. That's all I can remember about it.
So we talk about this one. The one that's responsible for the release is going to be, this is on the neuron still. It gets all the way down to they call it the axon terminal. And you have voltage-gated calcium channels. That
On the nerve.
I don't know why I didn't say it. I was like,
they let calcium into the end of the nerve, and the calcium interacts with the vesicles of the acetylcholine, which causes it to exocytose or release into the synaptic cleft, which is that portion between the nerve and the muscle. And then that acetylcholine goes and it binds to receptors. Those receptors are acetylcholine gated cation channels.
That's what I was thinking of.
Yeah. What's the name of the enzyme that breaks down acetylcholine inside of the neuromuscular junction? This is a test question. I promise this is a test question. The answer is acetylcholine esterase. The enzyme acetylcholine esterase is responsible for breaking down the acetylcholine inside of the neuromuscular junction so that we don't get continuous sustained contractions.
So that's the one that is responsible for preventing tetany.
Correct.
Okay.
Yeah. If we don't have acetylcholine esterase, then we would get tetany because the acetylcholine would just sit in the neuromuscular junction, firing, letting stuff away, letting stuff in and in and in and in, and we would constantly be sending a signal to the muscle.
Okay.
Which of the following is the correct sequence of events in muscle contraction? Calcium ions. So there's two ways going about answering these questions. If you feel like you don't know the material super well, you can read through each of the answer choices. Or if you feel pretty confident in it, you can write it down yourself and then compare. So, for example, the correct sequence of events in muscle contraction. So we're probably going to be like, okay, with the actual muscle contraction, I'm going to start all the way back from the motor neuron. So look at this. Let's kind of take a look and see. Because, okay, we have to go from the motor neuron. We go to the acetylcholine gated channels. Then it goes across the sarcolemma. It goes into the T tubule. We get calcium release. And then we kind of look and pick and see, look at the answer choices and see what works best. So calcium ions bind to troponin, myosin heads bind to actin, power stroke occurs, muscle relaxes. Okay, interesting. B, the action potential travels down T tubules. Calcium ions are released from the sarcoplasmic reticulum. The myosin head binds to actin, muscle contracts. That sounds pretty good. ATP is hydrolyzed. The myosin heads detached from actin. Calcium ions are pumped back into sarcoplasmic reticulum. Muscle relaxes. Again, we're looking at muscle contraction. So this is asking about this is talking about relaxation. So not that one. Acetylcholine is released from the motor neuron. Action potential generated in the muscle fiber. calcium ions bind to troponin, muscle contracts. That feels like it skips a whole lot of steps. So B, I think, is going to be our best answer.
Is that one that, like, if you were to ask that exact question, is that one that you would say you could kind of argue for D, even though it skips a bunch of stuff?
You potentially could, yeah.
Okay.
Like you technically could. In this case, with this one, like the main components of muscle contraction, is the myosin head binding to actin, doing the power stroke, all that kind of stuff.
Yeah.
So that's why like B is definitely the correct answer. Depending on the teacher and his mood and how you present it, you could probably get half points for D, is what I would say. Okay. As long as you go through like, oh, well, I was thinking from the whole sequence of events of muscle contraction.
Yeah.
And actually,
because I feel like with those answer choices, you almost have to combine B and D, and then you have the whole
potentially. Like, I would probably, as a professor, I'd probably argue, well, we're talking about muscle contraction, not muscle excitation.
Okay, yeah.
That's probably how I would argue that. If a student came up to me and was like, hey, but like this is the like said like the whole process, like this is how I fit it in, that's where I'd be like, if I was a professor, I'd be like, okay, yeah, you've demonstrated that you do know it. Here's some points back. I think that's all the questions I have currently. It is. I will make a whole bunch more questions. Maybe we'll do, what do you think, like 50, 60 questions?
Yeah.
And do something like potentially on Saturday.
Sounds good.
Sounds great.
Also, I have been not very good, and I've supposed to have been holding Thursday sessions for biochemistry 2. Is that something that we want to do? Is there a better time? Are people feeling overwhelmed with all this stuff going on? Would it be beneficial? What do y'all think?
Personally, I think Thursday biochem would be wonderful.
Yes, I agree. I am drowning.
I'm feeling a little nervous about biochem right now.
Same. I agree.
It's pretty easy for it to turn into a back burner class. Is that what y'all are feeling?
Yes.
No.
Yeah.
I have my one-on-ones with Sarkar, and I still feel like I'm drowning.
Okay. We'll go ahead and we'll schedule that in. We're going to do the same time, 6:30 on Thursday. Okay. We're going to plan on doing biochemistry 2. I think what we're going to do, we're just going to hit hard and focus on the Linus learning questions, because I genuinely think that that's the best way to kind of get through everything. If you're feeling like a little bit lost or anything biochem, I do have videos for biochem, at least for a little bit. Let me see how much I have in biochem 2. I have up to chapter 7 in biochem 2. So I think you guys are what? Are you guys chapter 3 right now? Chapter 4?
3, 4, yeah.
3, 4? Yeah. So I have up to chapter 7 going over the book in biochem. So if you need a little bit of review before Thursday, go ahead and hop in there. Otherwise, I'll see you on Thursday, and we'll get cracking on that, and then I'll set up a scheduled set time for either Friday or Saturday, depending on whatever it is feeling. Maybe Sunday potentially. Is your guys' test open all day Tuesday, or is it?
No, it's during during our lecture time.
Synchronous. Yeah.
Okay. Sounds good. So we'll plan. Monday is a holiday, so I'm not allowed to tutor on Monday. So we'll find a time for the weekend that we can do.
Cool. Sounds good.
All right, everybody, good work today. I will see
Thank
you guys
you.
later. Have a good
Thanks,
night.
sir. Have a good one.
Thank
Thank you,
you.
Drake.
Thank you,
Bye.
Drake. Bye.